Hi, folks. In science, as we're studying the cell, we're understanding that there's some unique functions for every part of the cell. Every part of the cell has a role to play, and we've discussed several of them so far. Tonight, we're going to look at chromatin, chromatin. This is this is a description for the chromatin. Chromatin is composed of a cell's DNA and associated proteins. Historic proteins and DNA are found in approximately equal mass in eukaryotic chromatin, and non-histone proteins are also in great abundance. The basic unit of organization of chromatin is the nucleosome, a structure of DNA and histone proteins that repeats itself throughout an organism's genetic material. Histones are highly conserved basic proteins whose positively charged character helps them to bind the negatively charged phosphate backbone of DNA. There are 5 histone proteins in the family: H1, H2A, H2B, H3, and H4. 2 H3 and 2 H4 proteins form a tetramer, which combines with 2 H2A and H2B dimers to form the disc-shaped histone core. Now, approximately 150 BP of DNA wrap around this protein structure almost twice to make a nucleosome core particle, with linker histone, and which is histone 1, H1, and linker DNA. This is called the nucleosome. So as you can see, these are structures that are important, very important to the DNA that's in the cell. Now, one thing I need to do real quick, is I want to go ahead and share with you. It's a little bit complicated, I know, but I want you to get as much as you can out of this. If you were to consider one of the structures, the chromatin it's like a chromatin fragmentation is a critical aspect of successful chip experiments. And so these chromatin are very important in the DNA that's part of the cell. Chromatin modifications. Chromatin is a highly dynamic nuclear protein structure responsive to a variety of endogenous and exogenous stimuli. In addition to DNA methylation, modulation of chromatin, conformation through various PTMs and histones, and non-histone chromatin associated proteins represents another critical epigenetic mechanism implicated in the regulation of gene expression. Genes are organized a certain way, and the chromatin plays a very important part in that organization. Now, when we're looking at a chromatin, when we're looking at a structure, what we have to remember is, and I was hoping to be able to find one. I thought I had a picture of it right here, but here is a picture right here that I can show you. It gives you a better understanding. The chromatin are located inside the nucleus. Chromatin is found inside. If you look right here at this right here, chromatin is right here. Chromatin is right in there. That's inside in the nucleus of the cell. And the DNA, as you know, is all about the genetic code. So what the chromatin does is, it says here, mechanistically, these modifications lead to the impairment of the interactions within and between nucleosomes, as well as to the recruitment of non-histone proteins. Proteins, non-histones, I know, technical stuff, but it's all a part of the DNA process. And enzymes, I think I'm going to just leave it right there. Well, we can talk about this more, but as long as you understand that it is a very key component, it's a structure that helps promote the DNA and keeps everything moving in terms of making the genetic code come together properly. And chromatin architecture in mammalic chromatin architecture and mammalian spermatogenesis undergoes extensive structural and functional reorganization, during which several tests specific histone variants and other chromatin proteins are expressed in a stage-dependent manner. So the chromatin is what we see, is what is part of the nucleus and deals with the genetic makeup of that organism. And it gets complicated, and for right now, that's where we're going to leave it right there. Thanks for watching.